Hello, and welcome to the next talk here in the eukaryotic session, and it's going to be Dr. Jana Uren, who got her doctorate from the University of Arizona and is currently an assistant professor in the Department of Agriculture and Biosystems Engineering. Jana focuses on the biodiversity, biogeography, evolutionary origins, and ecological roles of plant-associated microorganisms using next-generation omics tools to study microbial, symbiotic communities, and diverse lineages of land plants. Today, she's going to tell us about one of these small eukaryotes that interact with the plants, the fungi Xylariaceae. Please put questions in the Q&A box, and welcome, and thank you so much for joining us today.
Thank you, Jeremy. I'm really excited to be here to give this talk, and I really appreciate the organizers for inviting me to speak. So yeah, today I'm going to tell a little bit about a CSP project that we have with JGI on the genomics of Xylariaceae. So Xylariaceae are really common as endophytic fungi, and so for those of you who aren't familiar, endophytic fungi represent one of the most diverse groups of plant-associated symbionts. So endophytes form asymptomatic infections in the above-ground tissue, such as leaves and stems and bark and vascular tissue. Endophytes can also be found in roots. They are typically horizontally transmitted to their host through wind and rain. And so foliar endophytes in particular are highly diverse. And this is because they often or typically form highly discrete and localized infections. And so this is a diagram by Hara et al., where they took a 2 centimeter square piece of leaf tissue, they surface sterilized it and cut it into 2 millimeter square tissue pieces, and then they cultured endophytes from that. And what they found is that often each little 2 millimeter section of leaf held a unique endophytic fungus. And so really then these endophytes within these leaves create this mosaic. And so because of this, because of these discrete infections, endophyte biomass in the leaves is typically very limited. So it's difficult to do metagenomics or transcriptomic studies to study endophytes in planta. And so although they're cryptic infections, endophytes can play a variety of really important ecological roles. So for example, endophytes can protect plants against pathogens. And so in this experiment by Arnold et al. In 2003, in Theobroma cacao, they inoculated leaves with endophytes and then challenged them with a pathogen. And what they saw was that leaves that had endophytes did not have any signs of disease. In contrast, leaves that did not have endophyte colonization had lesions. And so I'm really interested in understanding both the ecological and evolutionary context of the endophyte symbiosis. And first, one of the most important things has been understanding where endophytes are distributed. And so, for example, in collaboration with Betsy Arnold at the University of Arizona and Francois Lutzoni and Jolanta Middlekowska at Duke, we've sampled in 12 different sampling sites across North America and Eurasia. And in addition to looking at how endophytes are distributed in these different geographic locations, I want to highlight that we're also interested in where endophytes are distributed in different hosts across these different geographic regions. So for example, in each one of these sites that are marked with a star, we sampled 20 phylogenetically diverse plant and lichen species that span these major lineages of land plants and lichen-forming fungi. And for those of you who might not remember what a lichen is, lichens are fungi that associate with green algae and with cyanobacteria. So for each one of these plant or lichen hosts, we surface sterilized the tissue. And in total, across all of these different sampling sites, those efforts have yielded over 16,000 cultures of endophytic fungi and across almost 800 different host individuals. And so each one of these cultures has been sequenced for the ITS and partial large subunit ribosomal DNA. And then in parallel with that, in our recent work across the boreal biome, we paired culturing with culture-free methods using amplicon sequencing of the ITS2 locus. And in this way, we're able to directly compare ecological inferences based on culturing and culture-free methods. But then importantly, the cultures themselves represent a reservoir of diversity that we can perform experimental assays and then also genome. And so from this work, we know that foliar endophyte communities as a whole are structured by their host lineage at both local to global scales. But the endophyte species themselves can display different levels of host specificity. And so the best way that I've found to look at this is in a network diagram. And so here I'll walk you through this diagram. So each of these large circles with the abbreviation is a host. So we have Magnolia phyta, Pinophyta, ferns, lycophytes, bryophytes, and lichens. And so then each one of the fungal OTU or species is shown with a smaller circle, and lines connect them to their. And so here I've colored the different OTUs by the number of host lineages that they associate with. And so red OTUs associate with only a single lineage, whereas OTUs that associate with more lineages have different colors. And then, so for example, the dark blue circles in the middle are endophyte species that associate with all these lineages. And so this is a network diagram of congeneric hosts across the boreal biome, so almost 800 OTU. And in this analysis, we found that overall about 24% of these OTUs, which is a proxy for species, that they were affiliated with only a single host lineage. What's interesting about this is that, so these are these red circles around the outside that appear to be host specific. What we found is that often actually these endophytes can be both host specific and specific to a particular geographic region. And so we really have these kind of endemic host specific endophytes. So in addition to understanding that ecological context, I've been working on trying to better understand who these fungi are and how are they related to closely related pathogens and saprotrophs. And so, you know, we can take ITS sequences and we can blast those, and that gives us an estimate of the taxonomy potentially of these species. But the majority of endophytes typically represent novel species, and they often have no close matches in GenBank. And so in order to understand where they fall in the tree of life, we use phylogenetic analyses. And so I've been focusing on understanding the evolutionary history of endophytes. And in particular, I've been focusing on this group of fungi, the Xylariaceae. And so in my PhD work, I isolated over 2,000 endophytic isolates that putatively we clustered into 92 different species. And so these were isolates that were collected across 5 different regions of North America. And one reason that endophytes in the Xylariaceae are interesting is because they are incredibly bioactive. So they produce a lot of novel secondary metabolites that have been shown to be important in agriculture, industry, and in medicine. And then in addition, the known described species of Xylariaceae are ecologically important. So the Xylariaceae is one of the largest and most diverse families of filamentous fungi. There's over 1,300 described species. And these species are very important as wood decomposers and litter decomposers, as well as being plant pathogens. And so they reach their highest species diversity typically in the tropics. And so as part of a NSF-funded project, I took these 92 putative species and incorporated them into a multi-locus phylogenetic analysis with all existing sequences of known Xylariaceae described species. And so this was almost 350 total taxa. And so I don't have time to go into detail about what we found with these analyses, but just briefly, I'll just say that, you know, by including endophytes within these phylogenetic analyses with these described species, we significantly increased the known phylogenetic diversity of fungi within this family. In addition, if you remember, each one of these cultures is linked to its host information, its geographic information, and in addition to some substrate information as far as living leaves or dead leaves. And so taking that metadata and looking at it in this phylogenetic context I was able to show that for most of these fungi, they associate with a very broad phylogenetic diversity of hosts, and then also that these fungi are widespread in different substrates, such as both living leaves and lichens, and in decomposing leaves, and sometimes in wood. And so that work really formed the basis for the CSP that was funded by JGI. And so in that CSP, on the comparative genomics of Xylariaceae, our goal was to compare, do comparative analyses to understand what are the functional roles of these endophytic fungi in the context of their phylogenetic placement, as well as their ecological information. And so this project was in collaboration with the co-PIs, Jen Wisecaver, who's at Purdue, and Betsy Arnold. And I also want to just highlight the contributions of Yuming Ju and Jason Slott, who contributed to this project. And without their help, we would not have discovered some of the cool things we found. So the goal of this project was to reconstruct a robust phylogeny of the Xylariaceae that contains endophytic, saprotrophic, and pathogenic isolates. The multi-locus analysis we did previously lacked backbone support, and so we needed phylogenomic approach to better resolve those deeper relationships. And then what we wanted to do was to examine the genomic factors that contribute to endophyte interactions. And then because of the bioactivity of Xylariaceae fungi, we focused a lot on the secondary metabolite gene clusters. And so we sequenced the genomes of phylogenetically and ecologically diverse Xylarian fungi. So we chose isolates to represent all major clades of Xylariaceae. And I'll just note now that there's some systematic revision occurring within this family, and I'll clarify that more, but it's kind of in flux and a bit of a mess. But we chose isolates across all major clades based on that multi-locus analysis. And then specifically, we chose closely related pairs of endophytes and non-endophyte species to the best of our ability. And in that way, we could minimize the effect of phylogeny when we wanted to compare what makes endophytes potentially different from non-endophytes. And so in total, we sequenced about 25 PacBio genomes, and then we did Illumina sequencing for the remaining taxa. And so I want to first highlight that all of the work that I'm showing today was done by my postdoc, Ernesto Franco. And so we sequenced the genomes of 88 genetically and ecologically diverse fungi. And the genomes ranged from 34 to 60 megabases and contained about 8,000 to 15,000 genes. So typical ascomycete genomes. In contrast to what's been observed for other symbionts, we didn't find an association between genome size and trophic mode. So endophytes did not have smaller genomes. We reconstructed the phylogeny of the family with about 1,500 universal single copy orthogroups, and that phylogeny is here on the left. And what we found was that that we got a highly supported tree showing two major clades. And again, with that systematic revision, instead of one family, it looks like we have multiple families. And so here, for the rest of the talk, the top clade is the Hypoxylaceae, and that's shown in blue. And then the bottom clade is the Xylariaceae plus members of the Graphostomatataceae. But I will refer to this bottom clade in red as the Xylariaceae sensulatu. And here you can see that the endophyte genomes are highlighted in this teal color. And so we have a mix of endophyte and non-endophyte genomes throughout this, of fungi. So because of the high bioactivity of fungi in this group, we used anti-SMASH to predict secondary metabolite gene clusters. And so consistent with that high bioactivity, we found almost 7,000 putative secondary metabolite gene clusters across all of these genomes, which is considerably higher that's been than other groups of fungi. And so we found the average number of gene clusters per genome was almost 72. And then just to put that in context, the average number across the Pizizomycotina is about 42. And so the number of secondary metabolite gene clusters was significantly enriched in this group of fungi. And that's congruent with the bioactivity that has been observed in chemical assays. And so overall, we found that the most abundant type of secondary metabolite gene cluster were the type 1 polyketide syntases. There were almost 3,000 of these, and there were almost 30 per genome. And so one thing that we found unexpectedly, actually, was that there were significant differences in the genome content between these two clades, the Hypoxylaceae and the Xylariaceae sensu-lato. And so in particular, we found that the Xylariaceae genomes shown in red were larger than the Hypoxylaceae genomes. And that this is associated with also an increase in the number of repetitive elements within the Xylariaceae compared to the Hypoxylaceae. And then in addition, Ernesto performed ancestral state reconstructions, and we showed that the Xylariaceae have experienced more gene gains and more gene duplications, but both groups have the same number of gene losses. And so interestingly, these patterns are consistent for both endophyte and non-endophyte genomes across these clades. So we see these major differences. And one reason we see these differences in genome size is that it appears that the Xylariaceae has experienced more horizontal gene transfer events. And here I want to highlight Jen Weiskäfer, who performed these phylogenomic analyses to characterize horizontal gene transfer. And so what we found was that most of the horizontal gene transfer events appeared to come from bacteria, but there were also genes from plants, viruses, related fungi. And then interestingly, although we haven't observed very many differences between endophyte and non-endophyte genomes at this scale, we did find that endophytes had more horizontal gene transfer events compared to non-endophytes. And that horizontal gene transfer is one reason why we see such an expansion in the secondary metabolite gene clusters. And so here I'm just showing a correlation between the number of horizontal gene transfer events per genome and the number of secondary metabolite gene clusters predicted per genome. And we can see that although Hypoxylaceae in blue has had fewer horizontal gene transfer events, that regardless that the gene transfer events are positively correlated with secondary metabolite gene clusters. So both clades have experienced horizontal gene transfer of secondary metabolites. And then the result of this is that if you look at the number of secondary metabolite gene clusters as a function of the number of genomes, and so here, for example, in a rarefaction analysis, that the Xylariaceae clade in red has an amazing richness of secondary metabolite gene clusters, regardless of the number of genomes that you sample. And so a significant increase, and here we're showing non-singleton secondary metabolite gene clusters in Xylariaceae compared to the Hypoxylaceae. And then the gray line at the bottom is the Sordaromycetes outgroup. And so this high secondary metabolite gene cluster exceeds that of fungi that are well known for their bioactivity, such as Aspergillus and Penicillium. So we wanted to compare these predicted secondary metabolite gene clusters to a database of known secondary metabolite gene clusters. And so we used, we compared our data to MyBig, which is a repository of known secondary metabolite clusters. And so what we found, so here I'm just showing, this is the phylogenetic tree on the left, and then each one of these columns is a hit to the MyBig database. And so overall, about 53% of the predicted secondary metabolite gene clusters in our genomes had a hit in MyBig. But although the similarity of those gene clusters varied, and so here the grayscale bar on the left is showing how similar those are. And then within this phylogenetic context, we can look at the distribution of those hits. And so what we find that obviously you have some secondary metabolite gene clusters like melanin biosynthesis or things that are have very high similarity and that are shared among most of these taxa. And then here I should just mention that the color coding of each column at the top is the taxonomy of the best hit. And so, for example, hits for our Xylariaceae or and Hypoxylaceae are, hitting to Cerdariomycetes, Leoshiomycetes, a lot of Uroshiomycetes, and some Dithydiomycetes, as well as just one or two bacterial secondary metabolite gene clusters. And so, interestingly, we found that across all of these hits, about 602 100% identical to my big accessions for known secondary metabolites. So, for example, alternariol, fusarin, each C toxin. And so these are toxins that have been shown to be important for virulence, for fungal virulence of plant pathogens. And so interestingly, we found them here, and they were found in both endophyte and non-endophyte genomes. And then Jen performed another analysis that basically showed that that a high number of these clusters are predicted to contain genes that may have been horizontally transferred. And so you can see that the little black marks at the top are hits that have a gene that is putatively horizontally transferred. And so the 100% similarity of some of these accessions is consistent with horizontal gene transfer of these clusters. It's just difficult to detect in phylogenomic methods because of the close relatedness of these fungi. And so one thing we wanted to look at, do endophytes differ from non-endophytes? It's kind of the million-dollar question, like how are endophytes different? When we looked at data across this entire across the entire family, we found few differences. But as I mentioned, we selected taxa that were paired endophyte, non-endophyte. And so we had 15 pairs of closely related sister taxa that were endophytes and non-endophytes. And we performed a statistical analysis to test the hypothesis that there was no difference in gene content between these endophytes and non-endophytes. And so here on the left, I'm just showing for Xylariaceae in red and Hypoxylaceae in blue. And values above 0 indicate that saprotroph or non-endophyte genomes had higher, for example, higher number of plant cell wall degrading enzymes. Interestingly, we found that for Xylariaceae, there was no difference between endophyte or non-endophyte genomes. This is pretty consistent across different types of genes, so peptidases, secondary metabolites, transporters. Again, we see differences in the Hypoxylaceae between endophytes and non-endophytes, but those differences are not visible in the Xylariaceae. And so that suggests that all of these horizontal gene transfers and gene duplications have really kind of erased that genomic signature of trophic mode, and suggest that potentially these endophytes are also capable of being saprophytes, for example. And so we looked at that by using mass loss assays. And again, so the y-axis here is the amount of mass loss of pinus needles. And again, we saw that, yes, those genomic differences result in functional differences where members of the Xylariaceae, regardless of whether they were isolated as endophytes or saprotrophs, are able to degrade more tissue compared to Hypoxylaceae. And because we have all of the host information, we can also look to see, is there a difference in how these fungi associate with different plants and lichens? And so what we found was that Xylariaceae in the red associates with a greater phylogenetic breadth of plant and lichens. So here that was measured with looking at the number of plant families or lichen orders that these fungal species associate with. And so it looks that, these genomic differences obviously are having an impact on the ecology of these fungi. And so then lastly, I just want to show that, interestingly, we found an association between the phylogenetic breadth of endophytes and the predicted number of secondary metabolites classified as NRPSs. And so it looks like, and this is just for endophytes in the Xylariaceae. So endophyte genomes with more NRPSs associate with a greater phylogenetic diversity of plants and lichens. So here, although secondary metabolite gene clusters are often known to be involved in pathogenicity and competition, this association suggests that there may be a role of these secondary metabolites in actually facilitating symbiotic associations with really diverse plants and lichens. And so in conclusion, We found that these genomes, regardless of clade or trophic mode, contain secondary metabolite gene clusters, and that over half of these appear to be novel. And then, although ecologically similar, so both Hypoxylaceae and Xylariaceae are endophytes and wood degrading fungi, we found really significant differences in the genome size and genome content between these two major clades, which was surprising to us. The genomes of the Xylariaceae sensu-latu are larger. They have more repetitive elements and more secondary metabolite gene clusters. And then this is potentially, we think, due to more horizontal gene transfer events of those secondary metabolite clusters. And we found that these genomic differences are associated with enhanced lignocellulose decomposition, as well as enhanced symbiotic associations across phylogenetically diverse plants and lichens. With that, I just want to thank really everyone at JGI, Igor, and everyone who's contributed to this project. It's a very big project, and I'm very thankful for everyone's help. Obviously, Ernesto, who's done all these analyses, and then members of my lab, the Wise Caver lab, collaborators at Duke and Ohio State, INRA, and then all of the F1000. So with that, I'll take any questions.
Thank you so much. That was really exciting and interesting to see what you can learn from vast sampling of these these endophytic fungal species. So there's a couple of questions. One of these is that is there any, based on your sampling, is there any kind of coevolutionary patterns with hosts? I mean, are these things moving in a specific direction through selection?
Yeah, that's a great question. And so I was originally interested in that when we did the multi-locus phylogenetic analysis. And what's really cool is that, so fungi have both sexual and asexual lifestyles typically. And so the fruiting bodies of the described species tend to typically associate with angiosperms. And if you look at only saprotrophs or only described species, it looks like they've specialized on angiosperms, especially in the tropics. But what we see when we incorporate endophytes and we have all this metadata for all these different plants and lichens, we find that that as endophytes, they associate with all these different lineages. And so when you throw that into the mix, it really looks like we don't see any sort of coevolution. So it's really interesting.
Ah, great. So then so can you just briefly explain how you identify the HGT, horizontal gene transfer events in the genome? And then are most of these commonly expressed in when you identify them?
Oh, yeah. So the way that we identified them was based on a pipeline that Jen Wisecaver developed called the Alien Index. And actually, it just looks at, within this phylogenomic framework, basically, how likely is it that that there's phylogenetic incongruence of those genes. And so Jen characterized that. And so sometimes in a phylogeny, it's very clear, right? You can see, oh, it looks like these have been horizontally transferred. And then other times it's a little, difficult to say. And so the data I showed are what we think of as almost 300, like very high confidence HGT events. Mm-hmm. And like I said, it's difficult once you start getting more closely related taxa. Right. And our phylogenetic analyses suggest that some of these genes are just moving around fungi pretty rampantly. Whether or not they're expressed, we have some data that shows that, for example, the secondary metabolite cluster fulvin has been transferred from Uroshiomycetes to Xylariaceae, and that product is actually made in culture. So it's an area of interest for us right now.
Yeah, so these HGT and the repetitive sequence both drive the differences in genome size across there.
Right, it looks like, they're acquiring genes from the environment and they're not tending to lose them, which suggests that they're important, right? So their genomes are kind of just getting bigger, they're keeping them. And I think we think probably that's because they're under selection for these different host environments. But then obviously there's processes of gene duplication that are also at work.
And did you mention that there's a higher HGT events in endophytic versus non-endophytic, or is that and
that was
on like why that would be?
I mean, we know that genes with ecological importance can be horizontally transferred and then they are maintained because of selection. And so I assume that the these genes that are being transferred have, provide the fungi with a selective advantage to interact with these different hosts. And so that's kind of our working hypothesis. I'd have to dig back and look and see, exactly what we found, but it's difficult to decouple the significant signal of phylogenetic history from this trophic mode. And so we really need to focus on kind of those paired comparisons of closely related endophytes and non-endophytes to tease that apart.
Great, great. Well, I want to thank you again for showing up today and giving a great talk and really opening up our eyes to this amazing diversity and exchange of material that's going on in these closely related fungal species here. And you can continue to take a look at the chat there and maybe answer a couple of chat questions. We've got a few questions we didn't get to today, but we're going to have to move on to the next talk. So thanks again. Appreciate it.
All right. Thank you very much.